Hi everyone, we hope you're all having a good start to the week. We're delighted that you've chosen to join us for this episode of our Exploring the Microbiome webinar series. Today we welcome Professor Anthony Buckley. Anthony leads the Microbiome and Nutritional Sciences group as Associate Professor in Gut Microbiology. His team uses nutrition to modulate the host microbiome for better health benefits, covering novel prebiotics, the detrimental effects of nutrition, ulcerative colitis, and understanding the gut-brain axis. His group collaborates with nutrition, pharmaceutical and biotherapeutic industries and academia. His talk is titled Developing in Vitro Microbiome Models of the Human and Porcine Gut. Please add any questions you may have into the chat box and we will answer them at the end. Thank you, and on behalf of Dr Buckley Scientific, enjoy the webinar.
Hello, my name is Anthony Buckley and I work at the University of Leeds as an Associate Professor in the School of Food Science and Nutrition. And my research is really looking at the human and pig gut microbiome, so this collection of microbes that live in our intestinal tracts. We use in vitro models to look at the differences in probiotics and prebiotics, so how nutrition and other microbes can really influence the gut health of different animal species. And it's a great pleasure for me today to talk to you about some new models that we've been developing here at the University of Leeds. One called RoboHog, which is a high throughput in vitro model, so benchtop model of the porcine hindgut. So I know it's me standing here talking with you, but it's really a fantastic collaborative interdisciplinary team that's come together to really work on these models. So I would like to extend a thanks to Professor Nick Kapoor and Pete Colmer and Dr. William Davis Birch, who are engineers by trade but have really been enthusiastic about this project. And alongside myself, Dr. Ines Mora, who's been helping develop the analytical pipelines behind the microbiome analysis that we do. And then especially on the RoboHog, Dr. Henry Greedhead and Katie McDermott, who really bring that background of animal biology and microbiome changes. So it's great to have them on board. Now, I know what you're thinking. RoboHog, great name, but why would anybody make a model of the porcine hindgut? Well, really, this is a huge economic opportunity for the UK. At the moment, the UK pork industry is worth about £1.4 billion pounds per annum to the UK economy. But there is a lot more where we can expand to, because we know that there is this mutualistic relationship between microbiome and host, and this occurs with all animal species, from pigs, fish to humans. But really in the pig world and farm world, there is these unique opportunities where we can try and tailor nutrition to optimize growth of the animal. We can reduce the feed costs. We can reduce the environmental impact of farming animals, but we can also try and improve the animal well-being as well. And strategically at the University of Leeds, we have a National Pig Centre here, which you can see in this picture just next to the M1 motorway there in the background. And it's a joint facility managed by industry and funded by the BBSRC, looking at how different nutritional products can really impact pig growth and pig welfare. But the problem here is that research is hampered in these areas, and that's really as a consequence of a lack of access to these sorts of facilities, a lack of facilities, and also the expertise needed in these facilities. But there's also cost implications as well. So this is where we felt that being able to provide a in vitro model that can sit on the bench and will really allow access to different researchers to move into this field and work on all of these areas. So when we started, we wanted to map the current in vitro technology, and we did this somewhat arbitrary by plotting them on this kind of axes where we've got experimental throughput versus ability to predict the in vivo outcomes. So in other words, how many samples or conditions can we run in an experiment at any one time, but also how predictive is that experiment of what might happen in the host. And on the low end of the scale here, we've got batch cultures, where these are typically plate-based batch cultures, or you can have a mini bioreactor arrays. And these are very, very high throughput, and they look at the short-term responses of, say, nutrition to a microbiome or a drug to the microbiome species. But these are very simplistic in terms of their batch. So when the nutrients are used up, then that's it. They're not controlled for pH, so they're not mimicking the in vivo conditions, and so therefore they're not particularly in vivo reflective. And then we can increase the complexity of these models to, say, single-stage models, and these can either be batch or continuous, where the media is continuously pumped in. And here we've started to introduce simple environmental controls, such as pH and temperature monitoring. But these lack the spatial temporal analysis that's needed when you're looking at animal biology. So these reactors might only mimic one portion of the digestive tract, for instance. And then on the other end of the scale, we've got these multi-stage models, and these are typically continuous models. They reflect the different gut compartments, and so they have very, very complex environmental control associated with them, but they're very low throughput because they're typically big and bulky. And so this is where we wanted to kind of move into, really, when we were redesigning these models. We wanted to increase the experimental throughput, but trying to keep the environmental complexity that's associated with really good prediction of in vivo outcomes. Now, in order for us to do this with RoboHog, we wanted to compare what was happening between a human and a pig, because we know that there are great models for humans out there, but what about the pig? So this is a very simplistic drawing of the pig digestive system, but one thing that you'll definitely notice is that the pig has this cecum environment here, and this is really this a fermentation bag, if you like, and this is because of the colonic fermentation of the fibers that they consume in their diet, and the fermentation that happens to break down those complex fibers. So what happens is that you get the nutrition or the ileal chyme that's coming through the ileum here, and then it will be stored in this cecum bag for a little bit before being expelled into the distal, proximal, medial and distal colon. There are also a couple of other physiological differences between pigs and humans. They have a warmer body temperature, core body temperature, compared to humans. They have a smaller pH gradient as well. So the pH gradient in humans, as you go from the start of the colon to the end of the colon, changes from about 5.5 to about 6.8. But there's less of a gradient in pigs, so from about 6 to 8.6. But there's also a retention time difference as well because of this cecum bag here. And so, but one of the key similarities is that both of these systems in the pig and the human are completely anaerobic, so they don't have any oxygen in them at all. And that really speaks because of the microbes that live in these different intestinal tracts are mostly anaerobic or facultative anaerobes. So they either can survive with or without oxygen, facultative anaerobes, or they're strict anaerobes and they really can't survive in the presence of oxygen. So this was another consideration when we were designing our model. Now, when we are creating this RoboHog technology, we decided on a triple-staged system here, and this is a continuous model still, because we wanted to reflect the continuous ileal chine that's coming through the small intestine into the cecum and the hindgut. And we wanted to have a look at the different compartments here. So this is how we've designed RoboHog, where we've got 3 individual compartments, and this first compartment is really replicating that ileum, cecal and proximal colon junction there. And the way that we designed this vessel, this first vessel here, is to really have a lot of dead space on the internal parts of this vessel, and this really mimics the cecum environment that can then be expelled and make its way through to the colon. And then we wanted to mimic the medial colon and also the distal colon in these subsequent vessels here. So what we've got is we've got media that's flowing through into the first vessel. It has an extended retention time because of this dead space, but then gets expelled into the second vessel, and then into the third vessel, and then out into the waste. And we wanted to mirror these physiological parameters in RoboHog that I mentioned before. So this pH gradient, there's temperature, but also because of the different ratio sizes between the vessel 1, 2, and 3, the retention time as well. Now, like all good microbiome models, we want to establish a complex microbiome in them. But to do that, we need a complex microbiome to start off with. So we use pig fecal matter, and we make a fecal slurry, a 10% weight per volume fecal slurry from this fecal matter, and we insert it into the H vessel to start the model, after we filtered, of course, because we don't want any blockages coming through. And this really allows us to establish the microbiome in these models. But in order for us to do this, we need to be able to keep this fecal matter in an anaerobic environment. And so this is how we do it, really, is by using our Don Whitley anaerobic cabinet. And you can see one of my students here, who unfortunately is about to prepare a fecal slurry in this environment. But this is a completely anaerobic environment, so it allows us to be able to pre-reduce our PBS mixture when we're making the 10% fecal slurry, and this allows us to get rid of all that dissolved oxygen that might be in there and replace it with an anaerobic gas mix. And then of course we are mixing this pre-reduced PBS with the fecal matter and then straining it to remove any large particulates. And so from here, what we can do is we can go into our model system now. And then this is where we wanted to really kind of validate our model system here. And to do this, what we did was we compared the microbial ecology of the fecal slurry input compared to how we recapture that microbial ecology in the model. And well, the way that we did this is by using bacterial taxonomic analysis by 16S sequencing. And what we can see here is, unless you're a really hardcore microbiologist, and I tip my hat to you if you are, you don't really need to know these names here particularly well. But what I really want to draw your attention to is the fact that this is the ecology that we've been able to establish in our from the distal colon of the pig. And this is the microbial ecology that we've established in RoboHog. And you can see that there is very good compatibility between the two models. We've got some differences, but by and large, we've got a very complex microbiome being established in this model. And we did this all the way through the different vessels as well, where we compared vessel 1 with the cecal content of a pig, and vessel 2, the medial colon, with the medial colon content of the pig as well. And so we've got a really good microbial composition. And the next thing that we wanted to have a look at is how stable was this microbiome here. So to do this, we have quantitative PCR reactions for 8 microbial species that are typically found in the porcine hindgut here. And what we noticed is that when we first start the model, we get some differences. The microbial populations take a little bit of time to stabilize, and when we think about it, this is a continuous model, so we're continuously getting nutritional mixes. And so what we see is an increase in some species and a decrease in others, but over time these stabilize, and when they do stabilize, just like the 16S data that I showed you before, they stabilize very similar to the input slurry, but also they're very stable in the model. Now, we don't get everything in the model, but what we are able to recapture is very stable, and we've been able to show this stabilization for about up to 4 weeks. We've never taken it longer than 4 weeks. So this gave us really good confidence in our model and what we're able to do with it. And now this is what we've been able to generate. So this is one rig of our RoboHog models. And what you can see here is you can see 4 independent model vessels, RoboHog triple stage vessels here, that are fed with this ileokine media that mimics the in vivo chyme coming through into the cecum and the colon. And as I said, these are all independent because they're all independently pH controlled, temperature controlled, the anaerobicity, the flow rate mimics the retention time. And if we wanted to, we could mimic different diets by changing this feed, or we can have 4 different feeds that feed each of these different models. And all of these environmental parameters are controlled by our controller box here, which instructs the dosing model to dose acid or alkali, depending on if a pH is going out of parameter. And so this is really quite a novel thing for in vitro model systems, because we are able to have high control over this environment, where we are streaming these measurements through the Internet of Things onto this Microsoft Azure database and able to display them using Power BI. But then also when we're looking at the microbial populations as well, we will be able to do quantitative PCR on each of these vessels independently. So we can sample vessel 1, vessel 2 and vessel 3 of all of these models all independently as well. So we get this spatial temporal analysis of what the microbial populations are doing. And this is just a really nice example to show you about how our live streaming profiles look like on our Power BI dashboard. And you can see here that we've got the different pHs, different temperatures, and all of the different reactors. This is an example from our human model, so you will see that there are some differences in pH and other physiological conditions. And then what we can see is we can start looking at the microbial populations on all these different reactors. So for instance, in this experiment, this is looking at Clostridia lepton species group, and in vessel 3. And we can see that they stabilize really nicely in each of the models. And then when we give an antibiotic, you can see how different they react depending on the dose of antibiotic that we give, and then how they stabilize or how they recover after the antibiotic. And we can do this with a range of different microbial species that we measure by quantitative PCR. But all of this is streamed as real time as we can make it. And then automatically uploaded into Power BI so that we can start looking at some of these analyses as the data is coming through. So this is a really nice feature for us. And this gave us a lot of confidence in the model in terms of what we're doing, and so we wanted to expand our capacity. Now, I'm not too sure what the collective noun of a collective of RoboHog models, but keeping the pig analogy, perhaps we've got a drift of RoboHog models now. And you can see here in my lab that we've got 12 of these models all running, or can we run simultaneously? So, trying to give you a little bit of data, and this is a really exciting thing that we've been able to do with our RoboHub models, because here what we wanted to do is we wanted to simulate the flow of antimicrobial resistance genes from farm to field. Now, obviously, we wouldn't be able to do this in the wild because we can't put antimicrobial resistance genes into the environment. So this was really nice for us to be able to team up with colleagues in the University of Leeds Geography Department and Earth and Environment Department here. And what we did was we were able to couple up these different in vitro models between the different groups that we have, so that we can then mimic an antimicrobial resistance gene that might be present in a pig, and how that might transmit through to the slurry. And then if a farmer applies this slurry onto the soil, how that might affect the soil ecology. But then also, if we grow crops, in this case radishes, in the soil ecology, what happens to the antimicrobial but also an antimicrobial resistance gene into here. And in each stage we did quantify the transfer of this transmission of this antimicrobial resistance gene and this antibiotic quantification. So what we did was we tagged an antimicrobial resistance gene. This was a CTXM beta-lactamase type antimicrobial resistance gene, plasmid based, and we put it into a Klebsiella pneumoniae pathogen. And we had 4 RoboHog models here where we didn't give any resistance gene. We gave resistance gene to one of the models only, and then we gave the resistance gene to one of the models, but then also gave 2 different antibiotics into 2 different models. And this was an antibiotic to drive the expansion of this antimicrobial resistance gene, so amoxicillin, but then also one that's used typically in farming, which is oxytetracycline. And here we then followed all 4 of these models and into 4 different slurry tanks, and then into the soil as well. Now, cut a long story short, this is essentially what we came up with. So when the antimicrobial resistance gene only was in our RoboHog model, we only detected it after 2 days, and then very little in the slurry. And then when we applied the slurry onto the soil, we was detected up to 12 days after the experiment started. However, when we added amoxicillin or oxytetracycline, we found that the antimicrobial resistance gene not only proliferated, but it also stayed in our RoboHog system longer, up until day 10. And then when we collected the slurry, we found that it was present at high levels in the slurry, interestingly very high levels in the slurry, with oxy given the slurry made from the RoboHog model where oxytetracycline was given. And then we were also able to detect this antimicrobial resistance gene in the soil 39 days after application. Now, fortunately, when we did grow the radish crop in these soils, and we looked at the radish root and the radish shoots, so the top and the bottom, we were not able to detect the antimicrobial resistance gene on this crop. But what we were able to detect is the presence of the antimicrobials, amoxicillin and oxytetracycline, in the radish root itself, so in the part that humans might consume. And that's really interesting. So just to kind of summarize this part, we have been able to generate a novel in vitro model of the porcine hindgut. We've looked at different pathogen carriage, and this could really lend itself to looking at the rational design of new therapeutics, for instance, new antimicrobials. We've been able to really push the capability of these models by linking them with other model systems to look at how we can measure these applications in a farm in vitro environment, and look at the AMR and antibiotic flow from farm to environment to food. And of course the next stage would be, well, how does that impact human health if we're consuming food and crops that's potentially containing antimicrobials? But also, we've looked at how nutritional trials can be run using in vitro models, such as RoboHog here, where we've looked at different novel feeds and also probiotics. And now we've just started a project to look at additives that can reduce the production of greenhouse gases. But there's still lots to do here. So RoboHog is mimicking the growth phase of pigs. But of course, when talking with our industrial advisors, one of the big problems of pig growth is this post-wean diarrhea. When the piglets come off their mother's milk, they typically would suffer from diarrhea because of overexpansion of opportunistic pathogens now. And so we want to be able to try and use RoboHog to be able to mimic what's happening in these piglets at this time. And that's quite an exciting phase for us to do. So I wanted to take a different tact here and tell you about some of the human systems that we've been using and been developing here. Unfortunately, I can't go back a slide, otherwise I would have told you, but this is my gut, and this is a project looking at different prebiotics, and this was funded by a company called Optibiotics and the Inform BBSRC OIRIC hub run out of the University of Reading. Now, Optibiotics is a prebiotic, and this is a fiber and mineral-based formulation. And this is really to drive a healthy microbiome support as we age. And it's the fiber and the minerals are really designed to target different bifidobacterium species from the proximal to the distal colon. And so our project aims here was to try and use MyGut, our human in vitro model system, which is the same technology as the RoboHog system that I've just spoken about, but we're trying to use it to gain a deeper insight into the effects of well biome in the different regions of the colon, because we want to be able to try and mechanistically understand what the active components are doing to see if we can try and improve prebiotics going forwards. So this was a really nice collaboration for us because it was a new academic industry application and using a commercial prebiotic already. But we wanted to try and understand the mechanisms of action behind well biome to see if we can enhance its effectiveness. But also, this really wanted to showcase the utility of MyGut to be able to support that technology pull through from academia to industry. So what did we do? Well, we took 12 models of our human system here, where we mimic the proximal, medial and distal colon of the human system. And as I said, they have slightly different environmental physiological conditions and parameters. And we took 12 of these models and we put in our fecal slurry from 3 different donors for our control models, but up to 6 different donors for our well biome models. And here what happens is that we allow the populations, the microbial populations, to stabilize, which takes about 2 weeks. And then we started to supplement into this first vessel our fiber prebiotic, and we did this for 2 weeks. And then we wanted to assess the robustness of any microbial changes that we've done as a result of this supplement. And we wanted to test the elasticity of the microbiome. What if we take the supplement away? And here what we did was we compared the microbiota profiles in the first vessel and the third vessel over time to give us that spatial temporal analysis. But we also wanted to overlay those microbial changes with any of their changes in the functional output. And so being fiber, one of the major jobs of the microbiome is to digest fiber, and they produce these short chain fatty acids such as acetate, butyrate and propionate. And so we wanted to compare the functional output in response to our fiber. So what does this really look like when we're doing our system? So as I said, we had 12 models. 6 of these models were given WowBiome, and then 3 of these models were also given each a fiber with low degrees of polymerization, so in other words, a low molecular weight fiber, and also high molecular weight fiber as well. We used 6 fecal donors, targeting healthy aging, so we targeted those people between 40 and 60-year-olds without any known GI disorders and had not consumed antibiotics in the previous 6 months. And this is really important for us because we want to get that complete healthy microbiome into our models. And so this is really what it looks like. And so with our first 3 fecal donors, we put them into 6 models, and in our first model 3 fecal donors, so that we compare directly the effects of WowBiome, our low molecular weight fiber and our high molecular weight fiber. And we repeated this with different microbiome donors so that we can really look at the individuality of responses to these fibers. But then we also selected a further 3 different microbiomes so we can get a really robust overall understanding of what's happening when different people receive WowBiome. And so here what we can do now is we can start looking at the spatial temporal microbial changes. So let me walk you through this graph because it is quite complicated. So what we've done here is once the microbial populations have reached steady state, we've been able to compare that level of the different microbiota species, as you can see here, as you can see here, and compared that to day 14 after we've given 14 days of fiber supplementation in our different models. And you can see the different colors here depending on which fiber they were given. These results then are the fold change of microbial populations changes compared to steady state. So steady state is effectively 0, and how much change have we induced in the different microbial species. And this is in our vessel that mimics the proximal region, so this first vessel. So one of the first things that you can see is that, as predicted, all of our prebiotics did increase bifidobacteria and prevotella in our first vessel, and this was very encouraging because that's what they're designed to do. But also what was quite interesting is that we noticed that there was a decrease in Enterobacteriaceae, and these are microbes like E. coli and Klebsiella and Citrobacter and Salmonella, so ones that are opportunistic pathogens. So that was quite encouraging to see in our first vessel. And then when we compare the different prebiotic fibers, we noticed that WowBiome was also associated with an increase in Bacteroides and Lactobacillus. And again, these microbes are very adept at carbohydrate metabolism, and especially Bacteroides. They've got a very enhanced repertoire of carbohydrate metabolizing genes, so they can take off and break down the side chains and the central chain of our fiber carbohydrate. And then when we also noticed is that the high molecular weight fiber, though, actually resulted in a reduction of Bacteroides species here. And then when we compare this with our distal region, well, the first thing that we noticed was that actually we saw a less of an effect in the distal region. And I think this is really important because when we look at, say, fecal samples in human studies, we assume that that's the total of what's going on. But don't forget, this is right at the end of our digestive tract. And so what's happening upstream might be magnified, or it might be completely different. So again, breaking down the different prebiotics, we were really encouraged to see that bifidobacteria and lactobacillus were enhanced all the way through the model when given WowBiome, and none of the other control fibers really did this, and so that was quite encouraging. And what was also quite interesting is that when we looked at our high molecular weight fiber, so a very branched chained fiber, we still saw this decrease in Bacteroides and Enterobacteriaceae, so that's quite encouraging. So these are really the spatial temporal changes that we can monitor in our model now, but we wanted to have a look at the functional changes. And this is where we got quite excited, because what we see is, in response to the fermentation of these fibers, we observed the fermentation of these fibers. They result in the production of short-chain fatty acids, which is acidifying the environment. And so therefore, to keep the pH within its set parameters, the model was dosing with alkali. Now, we were wanting to see if we can use this alkali dosing as a kind of measure, real-time measure for fermentation. And so what we wanted to look at here is this graph. And this is a graph over time, and this is what's happening in our model. So you can see that during steady state, we get some alkali added, we get some acid added. So the alkali is the blue line and the acid is the red line. But you see that we reach this steady state and what's happening here. And then when we started dosing with fiber, what we noticed is that there was changes. So the pH was dropping, and in order to keep the pH within its set point, the model was dosing alkali. And so what was happening here is that we were dosing alkali all the way through. And then remarkably, when we stopped giving the prebiotic fiber, the fermentation stopped, and the addition of fiber, the addition of alkali, went all the way down, and we went back to normal. So this was quite interesting. We wanted to see if we could use this region here to really measure whether there was any correlation between this area under this curve and short-chain fatty acid production. And so this was really encouraging for us. And I haven't got time to show you the short-chain fatty acid data today, but we saw really nice correlation between this area under the curve and the total short-chain fatty acids that were produced. And so when we look at this area under the curve and we can compare WowBiome and all the other fibers, what we noticed is that actually, regardless of which vessel we were in all of the vessels, we saw this fermentation shift throughout our model system. So increasing the fiber, and in this case, it didn't matter what the fiber was, we still got fairly equal total short-chain fatty acids produced. There were differences between the short-chain fatty acids depending on each fiber, but we were able to increase the fermentation as you go through the model, even into the distal colon, which is quite hard to do. So this gave us a lot of excitement, because now we were able to use our environmental monitoring to be able to correlate that with the fermentation potential of the different fibers that we might want to look at. So just to kind of summarize this work, we did look at WowBiome. We saw that it did outperform the compositional fiber controls that we used by enhancing the bifidobacteria and lactobacillus, not only in vessel 1 mimicking the proximal colon, but all the way through the model system as well, was suppressing Enterobacteriaceae. It was really encouraging that all of our prebiotics did increase this fermentation profile along the different colonic regions in our model, but it also gave us a lot of confidence that we could use MyGut as a tool for nutrition microbiome interactions, and this is what we really built it for, because we were able to get this spatial temporal variation between the nutrient and the microbes, and this really highlights the value of multi-stage models. We can look at the variation in response to the same prebiotic between different people, different donors, so we can start looking at tailored nutrition now. And we can use these environmental monitoring parameters as real-time measurements for fermentation. So I want to thank all of the people in my group, because without these, I wouldn't be here talking with you today. So I want to thank a big thank you to Monica, Bill and Will, who have been driving these first off, the creation of these models, but also using these models for their research as well. The excellent technical team that we've got here, Megan, my research technician, but also Miles and Joe, who've been instrumental in getting the lab up and underway. Our PhD students as well, as you can see here, who are all working on different projects, from prebiotics, probiotics, the interaction of pharmaceutical drugs, and also the interaction of kefir, for instance, with the gut microbiome. And so I want to thank Dr Buckley for inviting me to speak here, and for all of you for listening to me. Thank you.
Oh, hello everyone, and thank you very much for attending our webinar today. I'm Andrew Pridmore, and I'm going to be chairing the live question session for Dr. Buckley, our speaker. So thank you very much, Tony, for an absolutely fascinating presentation, and I'm hoping we'll get a few more questions. I'll remind everyone now to take the opportunity to type your questions into the box. And one has just popped up now. So somebody who said, is this study published yet?
No, so this is all unpublished work at the moment. So we are currently writing up the publication and the nutritional trial that we did with RoboHog. So the creation of the RoboHog models, but also the nutritional trial that we did with it. And the work that we've done with Optobiotics in our MyGut model is also being written up as we speak as well. So in fact, actually, that one's a little bit closer, and it should be submitted this month. So we will be looking to put preprints on these publications first. So they will become available fairly quickly.
Thank you very much. That's something we'll be looking forward to. I certainly will be one of the first to read it, I hope. So you've told us quite a lot of the technical detail about how these systems were developed. Are these systems accessible to other researchers? Will other groups be able to use them?
So it's a really good question, and at the moment we're exploring how we go about that. I think one of our main concerns here is that they're fairly new models. As I've said, RoboHog isn't even published yet, but we want to be able to just run a few more studies to be able to kind of gain confidence that when the technology is able to be disseminated, that's when we have full confidence in the system, and that gives everybody else confidence in the system as well. So we are exploring that at the moment. Of course, the university is looking at this from a commercialization point of view as well. And so we're currently speaking with them about how best we do this.
Yeah, of course. Thank you. Perhaps leading on from that, because I was commenting on how there were many different disciplines involved in developing this technology. Can you tell us anything about the challenges about working in such an interdisciplinary team?
No, I must admit it's been a really, really nice journey. Working with our engineering department, working with our animal biology department, and I think obviously there are challenges because it's different disciplines all coming together. So, for instance, communication and language that you use as well. It's best to try and keep things simple and explain things thoroughly. And what acronym might be used in one field is a completely different acronym in another field.
Yeah.
So it is quite nice. And I think one of the things that I felt, and this is very much my personality though, is that there wasn't. I felt that bringing a lot of these disciplines together, I don't have complete knowledge and control over all these different disciplines, but that's okay. As long as everybody explains what they're doing and what they mean, then we all get on really, really well, and the project accelerates. It's okay not to know because you're not an expert. I'm not an expert in fluid mechanics, for instance.
Yeah.
but it's great to be working with those, and it's one of those things that you kind of lose when you first start working with interdisciplinary teams, is that sense of control. But it does bring a lot of rewards as well, to be honest with you, Andrew. Um, I think that it's enriched my science, if I'm going to be honest. There are things that I thought I'd never be able to do, but now I've learned never say never, because if you get the right people in the room, almost anything is possible.
Absolutely, I would attest to that. If you work with other disciplines, it does enrich your science and your end results. Absolutely. Just another question I need to carefully read through here. Is it too early to use the increase in numbers of bifidobacteria and lactobacilli in the gut as a universal health benefit? For example, with IBM, and they may struggle with these fiber supplements even if it increases bacterial numbers.
Yeah, it's a very good question, and I think it's so these are all the environmental parameters that we've set up in these models are all healthy parameters, so therefore healthy individuals. Now we can adjust the parameters to see if we can try and mimic unhealthy states. For instance, we can try and mimic diarrhea by using a watered down version of the media that's flowed faster through the models, for instance, and things like that. Um, so but this is really in its infancy in terms of our research and our research questions for it. But I completely understand the question, because it's great saying, yes, fibers, you're increasing these beneficial microbes. But of course, fibers will also have different physiological
Yes.
conditions in different health status. For instance, IBS, you do get a lot of bloating and things like that. So it's always with kind of caution that we say this, and this is why we've kind of focused on a healthy microbiome point of view, and it's really for a healthy aging, because we do know that as we age, our microbiome functionality and diversity can start to decline a little bit. And we wanted to try and work with Optibiotics and Wellbiome to try and help try and promote that. And fiber in a relatively healthy everyday kind of person will cause certainly not as much bloating and pain as people would experience in IBS.
Yes. Okay, thank you. Yeah, that makes sense. Yeah. Talking of host factors, I guess I would ask, at the moment, your model system doesn't include any host cells or perhaps even the products of host cells. How do you think this might impact the response you're seeing in the microbiome?
Yeah, no, absolutely, and I think it's important to kind of really try and understand the limitations of model systems, because all model systems have limitations. Ours in MyGut or RoboHog is the fact that we don't have host cells, so we don't get host responses. For instance, normally when the microbiota produce short chain fatty acids, they'd be absorbed quite readily by the host cells, but they kind of accumulate in here. So there will be changes to the microbial response from the nutrition that's added. But we've got quite a lot of experience now looking at how predictive these model systems can be. Not only MyGut and these miniaturized triple stage models, but also other triple stage models that are out there on the market as well. And they've got very good both clinical and nutritional predictivity there. So one of the questions that we are looking to ask, because we've never been able to do this before, is can we individualize a MyGut model, for instance? And if we do that, can we mimic somebody who might go on a nutritional trial? So can we mimic that nutritional trial that person is undergoing with a model that's been primed with their fecal microbiome, and then mimic that nutritional trial? And we can try and use that to delineate some of the differences between whole systems like a human, but also model systems like my gut. So the short answer there, sorry, Andrew, is that we will see some of the changes because, as I said, you get this interaction between host and microbiota, and we simply can't mimic that at the moment. But rest assured, we're working on it.
Absolutely, thank you. Yeah, there was no need for a short answer either, because this information is all useful. I don't think we have any more questions coming, unless there are any people typing frantically. I think that's all of them. So thank you very much, Tony, for answering those and for presenting again. And thank you to everyone who's joined us today.
No, thank you very much for the invite. It's been a pleasure to speak with you all. And do contact me if you have any further questions about the results or the model systems themselves.
Yeah, of course. I don't know if your contact details are there for everyone to see, but if they can contact us, we'll certainly pass anything on too.
Yeah, they were at the start of the presentation.
Excellent, yeah.
So do feel free to have a look.
Okay, thank you.
Thank you, everybody.